Hello and welcome back guys, and in these videos we were talking about the bacterial cell structure. Till now we had discussed till that we had discussed about the plasmids, we have discussed about the capsule layers, and you can see what we have discussed all are labeled. If you have not seen those videos, you can see by clicking on the link which is there on this video somewhere. Okay, so now in this video we are going to talk about the cell wall. So we didn't talk about much about the cell wall during our previous videos. I told that it will be for some later video, and this is that video that is we are going to talk about this cell wall, which is the brown color structure over here. So this cell wall has been found of two different types in two different type of bacteria, and we have classified them as gram positive bacteria and gram negative bacteria, and as we will see. So let's start. Okay, so yes, so you can actually see the two structures of the gram positive and gram negative cell wall, and you can probably identify it. And if you cannot, this video is made for you actually. If you cannot, okay, so this one on the left of your screen is the gram. So let me change the, and let me enlarge the size. Okay, that will go. Okay, so this one is the gram negative gram negative bacteria, gram negative, let's write, and this one is the gram positive. So this is actually based on the bacteria which take gram stains. They are called gram positive as they take gram stain, and the bacteria which do not take is called gram negative as they do not take. So that is based on the gram staining procedure, which we are going to see later in this video. So you can see the general structures. Let me explain you this, what these structures mean. So this molecule you can see over here is actually the lipopolysaccharide. So let me write it down. So this one, this whole, okay, from here till the base, this one, this whole is the lipopolysaccharide, which the name suggests is actually lipid and polysaccharide, which is sugar. Lipopolysaccharide, such a large name, right? Very difficult to fit over here. Yeah, so that's the lipopolysaccharide layer. This actually consists of three parts, and this is the core part, this is the lipid part, and this is the polysaccharide part. Okay, so this one is the core part that is out of the scope of this video, but you can see that the lipopolysaccharide layer is present in the gram negative bacteria and absent in that of the gram positive bacteria. So that's a point of difference too. So now you can see that gram negative bacteria has two layers of cell membrane. So these blue layers are actually the cell membrane. So there are two layers of cell membrane. So if we if we are talking about the gram negative bacteria, then we should not have told that inside the capsule there is the cell wall layer. For gram positive there is the cell wall, peptidoglycan cell wall. For gram positive it is true, but for gram negative bacteria, inside the capsule there is the what do we say, cell membrane, then there is the cell wall, and then there is again the cell membrane. So that is the bilayered cell membrane. So actually the cell membrane is bilayered, and then that is two layers of cell membrane. Okay, and then this is actually the cell wall, which is made up of peptidoglycan, as I am saying many times. So let me label it again. This one is the cell membrane, cell wall. Cell wall. Okay, it is made up of peptidoglycan. In fungi it is chitin, and in plants it is cellulose, and in bacteria it can also be it is also made up of, and means simultaneously murine is also present, which is a mucopolypeptide, but usually it is said that it is made up of peptidoglycan. Okay, and you can see there is a space between these two layers called the periplasmic space. Now another difference between gram negative and gram positive, apart from this lipopolysaccharide and apart from it taking the gram stain, this gram positive taking the gram stain and gram negative not taking it, is the presence of this lipoteichoic acid. So these are also molecules like this. I have not drawn it like the in the molecular form by circles and circles. I have just drawn lines to represent it, and these are actually the lipoteichoic acids, and they are present in this cell membrane, inner cell membrane, and they actually perform the role to hold this cell wall into within the bacteria. Okay, so that is it. And we can, another thing this lipopolysaccharide layer does is that it plays a role in the immune system. So gram negative bacteria actually are more examples of gram negative bacteria are found which are infectious, which infect other animals, and therefore the immune system has found out ways to identify them, and therefore it identifies the sequence or the type of this lipopolysaccharide which is present in the cell membrane. Means what type of lipopolysaccharide is present. There may be some difference between the lipopolysaccharide present in one species of bacteria and in other species of bacteria, so that the immune system identifies, and then it can detect which species is attacking and take the steps after that. Okay, so that is another difference, that is the gram negative is more infectious than the gram positive. Yes, there are examples of some bacteria, gram positive bacteria being infectious, but there are few such examples. Another difference between the gram positive and the gram negative can be that these gram negative have more of the mesosomes. Okay, so this mesosomes I told about in the previous video, as you can see over here I have labeled it. So the mesosomes is actually the invaginations of the cell membrane formed due to effect of the antibiotics. So as I just told that the gram negative are more infectious, so if we study and therefore to kill it we will use more of the antibiotics, and therefore there will be more effect on it and there will be more prominent mesosomes, whereas gram positive bacteria have less of the mesosomes. So that was about the differences and the gram positive and gram negative bacteria. And now we are going to talk about the gram staining procedure. So what we do during the gram staining. So let's stay tuned and watch it. So here is it. Here is the gentleman which is Hans Christian Gram, and he developed first the gram staining procedure which we now use to distinguish the gram positive and gram negative bacteria. So actually he is a Danish bacteriologist, and he did this discovery on how to distinguish between gram positive and gram negative by an accident. Actually what he was doing is he was doing a biopsy test of the lungs of an infected person, and he was trying to identify the bacteria by using different types of stains. When he identified that this process can be used to differentiate between gram positive and gram negative. And on the right of your screen you can see how it looks when the gram staining is applied and the bacteria are seen under this microscope. So this violet ones you see over here are the gram positive bacteria, and the pink ones you see over here are the gram negative bacteria. Okay, so let us talk more about the gram staining process. So what we do in the gram staining process is take the sample of the bacteria we want to stain, we want to know about, and heat it so that the bacteria is actually fixed to the slide on which we are taking it. So this is important because when we do the process further, we use alcohol and we use the dyes, and this may remove the bacteria, or the bacteria may flow away with these. So to prevent that from happening, we actually heat it not at very high temperature, because it may damage the cell wall which we are interested about and may also kill the bacteria. But we heat it, and some bacteria do die, and the bacteria are fixed to the slide. So first step, let us write over here, is to heat and take the bacterial smear. So that smear is the bacteria which is fixed to the slide. So bacterial smear we take, bacterial smear. So we took the bacterial smear. Now what we will do, we will dry it in the air, and now we will stain it with crystal violet, which is a type of stain used in which has many applications, which has many uses, and one of the uses is this, the gram staining. Okay, so let us write over here, stained with crystal violet. So actually, to give you an idea about what the molecule is, I have just taken the images from the Google. So you can see this molecule on the right over here is the crystal violet molecule. 3 benzene rings, and there is the nitrogen over here, and this nitrogen, actually the top one, actually it is shown, this one contains a positive charge where this chlorine negative ion is attached or by ionic forces. So that is the crystal violet, and it is not actually required until and unless you are doing some very high level studies. So that is the crystal violet, and when it is stained, actually let me go back to the previous one. When we apply crystal violet, actually the crystal violet can penetrate this cell wall actually. So as you may know that cell walls are completely permeable in plants also, and in bacteria in this case also. So it can penetrate the cell wall, but the cell membrane it cannot penetrate very fast or very easily. So when we put the crystal violet in case of the gram negative, it first of all it sees a layer of cell membrane. So when it faces a layer of cell membrane, what happens actually is this violet gets trapped here at some places, and this trapping is encouraged by adding iodine in the third step. So here it is trapped, and in case of the gram positive bacteria, the cell wall is completely permeable, so it passes through here and is trapped over here. Some goes inside the cell also, but most are trapped over here. Okay, still no well and good. So stained with crystal violet, and then we actually see the black bacteria turn into bluish purple color, and then we wash it with water to remove the extra stain. Okay, so the required amount of crystal violet gets absorbed into the bacteria, and the extra crystal violet, we do not put it measured, very accurately measured crystal violet, we actually put some drops and wash it with water to remove the extra drops. So actually then we wash with H2O, sorry, the color is crystal violet now, wash with H2O. Okay, so we washed it with H2O, and now as I said, we are going to put iodine into it. So what this iodine is going to do is that, you can see the structure again. So this positively charged has had the chlorine before, and now this iodine will either form I minus or will form I3 minus, and then form ionic bonds, actually which are more covalent than the previous case, with this structure, which is with this crystal violet structure, and then form a huge crystal violet complex, CVI complex, crystal violet iodine complex. So you can see over here this crystal violet, and if the I3 minus get attached to it, the chlorine was a very small molecule, okay, so it could easily enter this cell membrane mesh. Now when the iodine is added, this reacts with the crystal violet and something like this occurs. So let us say that these small dots are the iodine, Okay, so wherever the crystal violet is there, the iodine will add to it. So here you can see I have drawn the iodines over here, and then the huge CVI complex cannot further escape from it. So it had entered one by one. The crystal violet entered, then the iodine entered. Now when they are fused to each other, they cannot escape it. So the third step, if I have not written, let me write down that addition of iodine is the third step. Iodine added, and usually it is a 0.5% solution. For your extra knowledge, I am telling 0.5% solution is taken. Yes, so that is the fourth step. Okay, now when we have added the iodine and all this is set up, we will wash it actually with either acetone or ethanol. So what this acetone and ethanol will do? So when we wash it with acetone or ethanol, these structures or these chemical compounds are actually very good at dissolving this bilayer, lipid bilayer structure. So when we wash it, this lipid bilayer will get dissolved and then will go away. But when we wash it with this, in this the gram-positive case, there is actually the cell membrane layer, cell wall before the lipid bilayer structure, so it will not affect. If some enters also, as the cell wall is completely permeable, it will enter and then it will not be able to completely wash out. Okay, so to wash it, it has to enter and then dissolve and then go out, which is very difficult process. But here the complete will be removed. So you can see most of the crystal violet complexes are removed. So what happens after we wash it with ethanol or acetone is that the decolorization takes place in case of gram-negative bacteria. So we will mention wash with alcohol, or acetone can also be taken, alcohol. Okay, so when we wash with alcohol, the gram negative gets de-stained, and then we could use now up to here it is enough to distinguish between the gram positive and gram negative. But how will we see the gram negative bacteria under the microscope? Because at this point of time this stain is completely removed, and to see a bacteria under the microscope it must have some stain, right? So we have to add another stain to it, but it must not be the crystal violet again, because if it is crystal violet, then we will not be able to distinguish between them. So now this is washed with ethanol. So let me remove it. Now this is existing in the gram negative bacteria, and now we will add, actually most of the times that we add saffron into it. So this is the structure of saffron for your extra knowledge, but it is not very much required in this video. So now we add saffron in. Add saffron in, or I think it is 1F only. Okay, so add saffron in, and saffron in actually is a pink colored dye, and in the place of saffron in we can also add the fuchsin or carbol fuchsin. Actually it is it has a darker stain, so it is advantageous, but people don't prefer it. People usually add the saffron in to it, and then we can see both the slides under the microscope, and then we will find out which one is gram negative, which one is gram positive. So hope this video was helpful. In the next video we will talk about the bacterial flagella and cilia, flagella and pili. So stay tuned and like this video if you like it, and please subscribe. Thank you.